I am Jing Zhang, assistant professor in the computer science department at UC Irvine. So today I'm going to talk about our computational efforts to dissect the gene regulatory grammar at a single cell resolution. A little bit more about my lab. So humans has a huge genome, 3.2 billion base pairs. And compared to the reference genome, we know there are many, many variations. And those variations make each of us one of a kind. But unfortunately, they also encode a lot of the risk for genetic disease. So my goal is to develop computational methods to interpret these variations in the human genome and link them to the phenotypes. And there are many different ways to look at the same problem. So my special way is to look at the gene regulatory grammar, and we see what dysregulations has happened that can cause those phenotypes. So to achieve this goal, my lab has been working on a lot of machine learning applications on genomic data modeling, ranging from like single-cell high-C data, single-cell ATAC data modeling, and spatial multiomics data modeling. And so we have been trying to provide some single-cell analysis toolbox to cover key regions of single-cell epigenome data processing. It ranges from the data analysis, basic data analysis algorithms, for instance, clustering, simulation, batch effect correction, integration, and some network construction to the very phenotype level, and try to apply those methods in the context of different disease. So we have developed different tools, but today, due to time limit, we are going to focus on several tools. So in this talk, we are going to focus on providing an end-to-end AI solution on genomic data modeling. And the first part of the talk is to talk about the data processing. So basically, how do we deal with this noisy data? And the second part of the talk is going to focusing on, after we clean up the data, how do we extract the knowledge that can help us to interpret gene regulation and help us to investigate disease for humans. And at the very end, we are going to briefly introduce some of our recent work. So let's go to the first part, and how to clean the noisy single-cell epigenomic data. So starting from 2020, we started to work on single-cell ATAC data, and it actually measures the open chromatin regions in each single cell. It has a lot of applications. For instance, it can help us to characterize cell states, to build the cell trajectories, and to investigate gene regulations. But unfortunately, this type of data is very noisy. Basically, we were given almost a binary matrix, right? With the rows represent cells and the columns represent different regions in the genome. And this data is very difficult to deal with because it's very noisy. It's sparse because we only have very limited DNA to profile the open chromatin regions. And we in the 3D genome, those features actually interact usually in a nonlinear way. So how do we deal with such data? So in every pipeline, when we are analyzing the single-cell ATAC data, a key step is basically dimension reduction. For those high-dimensional features, so we need to project it into a low-dimensional space before we are doing the clustering and many of the other downstream analysis. And before we started to work on this, there are many existing methods, such as PCA, NMF, autoencoders, and different methods. And many of these methods, no matter they are linear or nonlinear, they actually work very well in a single sample analysis. But in reality, we seldom did one sample analysis in a disease study. Right? So what we see is we usually sequence multiple samples, and we need to jointly analyze those samples. And then At that stage, there are a lot of issues in the data analysis. For instance, this is the early stage of data generated by two ATAC sequencing machines. You will see if we follow the traditional data dimension reduction methods, these two colors actually represent two sequencing technologies. And those data will be separated purely based on their sequencing technologies. And that's not what we want, right? And you may say, okay, how about we don't use different sequencing platforms to generate the multi-samples? And then we have no such batch effects. It's easy for us to analyze the data. Actually, that is not the case, because we know there are many natural confounding factors that can impact the harmonization of such data. For instance, as we are aging, our chromatin get looser and looser. So samples with different age, their accessibility profile looks very different from each other. So even you collect the data from, for instance, the same brain bank, you use the same sequencing depths, you use the same sequencing platform to generate the data, you still have a tremendous challenge to harmonize such data. So we started in 2020 to look at different methods to harmonize the multi-sample, like single-cell ATAC data. And so for our method is we start from the variational autoencoders because they can handle the nonlinear interaction of the high-dimensional features very well. And we want to extract the ideal cell representation Z that only represent cell states instead of different cell types. So cell states and different cell types. But in reality, if we use the vanilla version of variational autoencoders, the cell representation Z is usually incorporating cell states information and the confounding factor information. So our intuition is very straightforward. How about we try to force the cell representation Z is independent of the known confounding factor C? How do we do that? Of course, we can use GAN, but it's difficult to train. And many experimental labs, they told us they really don't like GAN, because of the training difficulties. And then what other options we have? So what we did is we introduced the invariant representation technology, representation learning technology in AI to this field. And we modified the objective function by add another penalizing factor. We calculate the mutual information between Z and C and add it into the objective. So with this methodology, and we are encouraging Z to be independent of C as much as possible. And the result is actually very encouraging. If you look at this data, we applied our method on a mouse atlas data with around 81,000 cells. And you can see the first comparison is our method, Sailor, versus Snap Attack at that time. And you can see actually the neural network architecture allow us to capture the nonlinear feature interactions pretty well. You can see different tissues can be separated fairly well. But the important thing is, if we compare our method as with the vanilla version of variational autoencoders, as highlighted by these two boxes, you will see the vanilla version of the variational autoencoder without the invariant representation learning, they have this L or long-shaped clusters. And we check why they have such shaped clusters. So we embed different sequencing depths on each cell, and the low use a deeper color, and the high sequencing depth cells using a lighter color. And you will see the vanilla version of variational autoencoder has an obvious artifact, which means within each cluster, for one cell's neighbor is the other cells that has been sequenced similar deeply, which mask the true biological signal at all. Because we want to define the neighbors only based on the biological similarity, right? So you will see in our method over there, within each cluster, these high and lowly sequenced cells, they were harmonized relatively like equally as compared to the vanilla version of variation encoders. And that's all because we made a tiny modification in the objective function. And later on, we also applied this method on a challenging case that has been sequenced on sequence data generated by two sequencing platforms. So after harmonization, you see these two colors were distributed relatively equally, like within each cluster. And if we look at the canonical marker genes, actually they like each separate group, which is harmonized well, can have a very high expression of canonical neural marker genes, which means with this simple modification, our data can be harmonized well. So this is our first effort in 2020. And then in 2021, we started to work on single cell multimodal data. And that's because we know for the ATAC data, they are not that effective to capture different cell types. So our collaborators started to generate a lot of single cell multimodal data. You will see, this is what we have. And we extended the variational autoencoders from one modality to two modalities. But we don't require the cell representation from RNA and the ATAC to be exactly the same because there is a chance that they are not synchronized. Instead of forcing them to have the same they representation, what we did is we train batch by batch. For each batch, actually, we are trying to keep the same cell-to-cell similarity from these two modalities. And for here, in other words, we are using a soft alignment for these modalities. And the second point is we know some cell types actually ATAC works better, although it's relatively rare, but we are going to show the case later, right? And for many cell types, actually, the RNA works better to separate them. So our thinking was if we are forcing a noisy data to be equally integrated into a high quality data, then you may not end up with the best result. So instead of weighing these modalities equally, we are actually assigning different weights according to the sequencing depth or other QC parameters, right? And we are trusting these different cells using different weights. And the third one is we inherited the invariant representation learning scheme from our single modality analysis. So we call this method CGSI. Here is one example that has been applied our method on the public PBMC data. As you can see on the top are our methods using one modalities, either ATAC or RNA, or increasing or integrating both modalities. The first RNA modality, actually, you can see that's two subtypes of T cells. RNA has some ability to separate them, but not the best. Actually, in that rare case, ATAC can separate those two cell subtypes of T cells relatively better. So by this reweighting scheme in our CGSI methodology, you can see these methods can be separated. These two subtypes can be separated better, right? And we also have a subtype of B cells that actually, by this reweighting scheme, they can be betterly separated. And here is the comparison of our methods with the existing methods. You can see by our clustering methods, the marker gene scores, and the motif enrichment for canonical marker genes are significantly higher as compared using other methods. So it shows that this reweighting scheme is kind of important. Soft alignment is also kind of important when we are aligning these two modalities. And later on, we are starting to thinking, okay, what can we get out of this single-cell multimodal data? Yes, we can project them into the same space. We can align them. And then what? What biological questions can we answer, right? So we started to work on this cross-translation model across the different modalities so that if we modify one modality, we can see the consequences in the other modalities. We can link the chromatin accessibility to the RNA status, which is kind of valuable, right? So what we did is we inherited our existing architecture, and you can see we have the RNA variational autoencoder and the ATAC variational autoencoder. So in our function, in our optimization function, we first trained two modality-specific variational autoencoders. In the second step, we let the RNA modality to go through the RNA encoder, and we get its latent representation. We train a neural network to project it into a different modality, right? After we project it in a different modality, we then protect it back and use a decoder function to reconstruct the RNA. And then we are connecting the RNA with ATAC already. And similarly, in the ATAC modality, we did the dimension reduction. We projected to a different modality, and then we projected back and we reconstructed. So we added this cycle-consistent training process to allow the translation of these modalities in the latent space. And we kind of forced them to harmonize within each modality. So we use adversary training to make sure that we are projecting the same cell type. They can't be separated easily, and so they are harmonized. And at the very end, we also added the invariant representation learning to overcome the batch effect. So this is our overall objective function. So we again applied our methods on this public PBMC data. As you can see, here is the marker genes. And we compared the originally observed RNA data, and we generated the RNA data from the ATAC modality. And their marker gene scores actually are very consistent to each other. And we extend our analysis from the marker gene scores to marker genes to all genes. As you can see, in most of the cell types that we have enough training data, actually the projected RNA expression is very similar to what we see, which means after connecting those two modalities, we can easily fill in a matrix, right? If we have missing modality, what is the other modality? And we can conduct easily some perturbation study to link different modalities together, right? So that is our three efforts to deal with the single modality, single cell epigenetic data, multimodality, single cell data, and try to make sense out of them, right? So after cleaning those data up, we are trying to extract some biological knowledge that may be important for biomedical study. So our first try is to construct the gene regulatory grammar in each cell type. What should we do, right? Again, this is the work we started in about 2021. And we have single cell multimodal data. And we have two very, very sparse matrix. X is the ATAC matrix, and Y is the RNA matrix. We are trying to see which element of the open chromatin region is a true cis-regulatory region. So surprisingly, actually, less than half of the open chromatin regions are really regulatory regions. I mean, there can be enhancers, silencers, others have other functions. Not every open chromatin peak is an enhancer. That's number one thing that we learned, right? But this is essentially a very challenging task. Why? Because except these two very noisy matrices, we have nothing else. In bulk tissue analysis, we have Hi-C, we have MPRA, and we have ChIP-seq. We have all kinds of cool functional genomic sequencing assays to help us. But here, we have nothing at the single cell level. I hope there will be new technologies yet. Cut and tag is coming out. But at that point, we don't have large-scale functional validation data at a single cell level. So what should we do? Again, our thinking is very straightforward. Let's look at one gene at a time, right? And we have the gene body, which is in yellow, and the promoter region, which is in gray. And we actually defined a gene neighborhood that's focusing on the promoter region. We go upstream and downstream 500 kb. You can change this region as you defined. And then to overcome the sparsity of the single-cell epigenomic data, we actually defined metacells, which is actually a widely used schematic in single-cell data analysis. And now that we have two dense matrix, and Y is basically the gene expression, now is dense already, and we have different X vectors to represent how open or close in this open chromatin region. And then the task is easy. We can build a predictive model between X and Y. And to handle the nonlinear feature interactions, we actually use a gradient boosting machine here. And after we have done this, we do model selection. We remove those that has no impact on gene expression, only keep those that has a high impact on gene expression to define them as cis-regulatory elements. Then we can impute the footprints in these regulatory elements, so we link the TFs to different genes to build the gene regulatory networks in a tissue-specific manner. And we applied our data, again, on public PBMC data, because at that time, that was one of the very few available data set. As you can see, when we are characterizing the open chromatin regions into three categories, high confidence are those that significantly impact the gene expression. The non-CREs means they almost have no impact, and the middle category. You can see actually the high confidence CREs showed the best power to separate different cell types or cell states. And the non-CREs actually almost has no power to separate different cell types. They are open probably for different reasons other than being the enhancer or silencer. And also we used independent Hi-C data to validate our results. And you can see this is the independent ChIP-seq data. That's 3K27AC ChIP-seq data, which is a canonical marker for active cis-regulatory regions. And we compared the ChIP-seq signal enrichment across those three categories of CREs. As you can see, for the colored lines, the top line are all high confidence CREs. They showed the strongest overlap with the cell type matched but independent 3K27AC ChIP signals. And we also overlapped with the whole blood ChIP-seq signals. You will see that's gray lines in none of the categories. They showed strong enrichment. So that gives us two messages. The first message is cell type specificity is important. It's important to use single cell data in different cell types because they look so much different across the whole tissue level function genomics. The second lesson is not every open chromatin region is a cis-regulatory region, and it's important to pick those that are really functional in terms of transcription regulation, right? And that's our second work. And I also will briefly introduce another work that is how to use the single-cell RNA-seq data to determine the cell phase. So single-cell RNA-seq data nowadays is very affordable and is a mature technology. But one problem is it's only give us a snapshot of what's going on in a cell, right? But sometimes we are very into developmental, analysis, and what we want to see what's the transcription process is going on in some amount of time, right? So on this task, actually, some methods has been used. For instance, we can infer the velocity of the RNAs, showing in a short amount of time how long it can change. And we can observe the gene expression data directly for a certain time point from the RNA-seq data. And then existing methods basically use linear methods to project between these two vectors. But then when we start to look at the problem, we always have a question. So why should we assume there's a linear relationship between the gene expression change versus the existing gene expression profile? For instance, two transcription factors can be required at the same time to activate a gene, which is actually obviously nonlinear. So how should we deal with it? So what we did is we developed a method that is called deep value. That's a neural ODE-based method to model cell transitions. So the X is basically the existing gene expression profile, and Y is gene expression, the RNA velocity, the change of gene expression. And we assume that they are sitting on the low-dimensional space. And we are using a neural network instead of a linear matrix projection to connect those two components. So, and then with this connection, we can actually make like a smaller step, like predictions continually to model this trend, this cell state transitions. So it has different applications. For instance, it can help us to model cell state transition. Another important thing is it can find the critical cell index. We call it a CGSI. So imagine that you are taking a roller coaster. The highest point of the roller coaster is actually a very important point because you know in a short amount of time, you're going to experience a high velocity change. Right? So this is similar in cell development. I'm not going to introduce the details due to time limit, but you can see our CCI index can help to identify such key cell developmental stage very accurately. And in the end, actually, we can find the developmental driver genes and distinguish them with the developmental passenger genes using permutation study. Right, that's a third application of our deep value methods. And now we are also into many disease studies. For instance, we are into psychiatric disorders and alcohol usage disorders and PTSD and stress responses in the human brain. We have collaborated with experimental labs to sequence over 100 brain samples, both RNA and ATAC, and we defined detailed subtypes of brain regions in the prefrontal cortex in the first stage, then extended in 10 different brain regions. And we find important differentially expressed genes, and then we actually find the cis-regulatory elements and the key variants that are regulating those DEGs or GWAS genes, trying to provide a mechanism explanation of these disorders. And I want to thank my collaborators who generated the data, also the whole SCORCH consortium, which give us a lot of valuable data to conduct our research. And thank for my lab members and my funding agencies. Let me know if you have any questions. Thank you.